Hello, my name is Linda Allen. Today is Tuesday, April 25th, and I'm with you at the Chicago McCormick Place Convention Center, and I have the pleasure and honor of interviewing Dr. Howard Winet, who has been a long-term member of the American Physiological Society. He's been affiliated with the University of California, Los Angeles, and his research has focused on bone healing and intravital microscopy. Welcome, Dr. Winet. Thank you for spending your time with us this afternoon.
Thank you.
If you're ready, I'd like to ask you a few questions.
Okay.
You grew up not too far from here, and I understand that you grew up on the west side of Chicago. Can you tell me a little bit about your growing up experience here?
Yes, I grew up during World War II, and about 3 years after my birth, my parents were divorced. And as a result, my mother had to go to work, and she sent me to a day and night nursery during the week while she worked, and then I'd either go to her house or I would go to my grandparents' house on the weekend. And that went on for about 4 years. And something happened which I did not ever get the details for. And I ended up with my grandparents, which were my father's mother and father. And I did not see my mother again for many years. Although she wasn't dead, or there was nothing serious that happened to her. But so I lived with my grandparents. And my grandparents were Orthodox Jewish. And my grandfather, who had grown up in Minsk, in a shtetl where he was lower class, he had always had the dream of being a rabbi, but he couldn't because he was from the lower classes. And I was the oldest of all his grandchildren, and he had some ideas of me possibly becoming the rabbi he never could have become. Unfortunately, as a youth, I wasn't very receptive. I was kind of disruptive in school. I was a bit of a problem, and I didn't study very much, and they had to call in my aunt to school and so forth. And I think I was probably an ADD.
Oh.
But I finally met in the third grade my first saving teacher, Mrs. Sheldon, who was able to somewhat calm me down. She took an interest in me. She got me into a camp during the summer, and I started to calm down a little bit. Now, at the age of 11, my father, who had been stationed in the Pacific, he was in the Army Air Corps during the war as a medic. He had been stationed on the Pacific coast, and he started a life there. By then, he had come to his third wife.
Oh.
Okay, and he sent for me to come to California. So at the age of 11, I left for California.
Had you seen him at all during this interim?
Yes, he had visited.
Okay.
He had visited a couple of times, but he was mainly in California. He was working in the airplane industry, building airplanes at Hughes in North. What was the name of the other company? Northrop and Hughes. And then he called for me, and I took a train. And
At the age of 11?
At the age of 11, there was a lady that was with me that was my guide. I remember stopping in Santa Fe, New Mexico, or actually it was Gallup, New Mexico, and it was quite an experience. And we lived in Santa Barbara. I don't remember too much about school at that time. And as yet, I had still not met any role model that had any academic leaning in my youth up to this point. And it remained so. About a year later, when I had just about become 12, my aunt died of lupus, my grandmother's daughter. And my grandmother was so upset by it that her family induced my father to let me go back to Chicago to console my grandmother. So at the age of 12, I returned to Chicago. And my grandfather, of course, thought this was a great opportunity because if you know anything about the Jewish faith, 12 is when you're supposed to start training for your bar mitzvah. And he, I'll bet he was of two minds when my aunt died because the prospect of me returning to Chicago was poor until that happened. So I went into training and I completed my bar mitzvah and that went okay. And then I was picked up by my father right after the bar mitzvah and we drove back to California.
Were you
Santa Barbara.
surprised by that? Were you expecting him to pick you up?
Yes, because it was sort of, I was on loan.
Okay.
Until my grandmother could calm down, after the death of her daughter. And so I went back to Santa Barbara again, and he was on his fourth wife then. She was probably the closest thing to a mother that I had. Because I spent more time with her than I spent with my biological mother. She was Mexican-American.
Oh.
She had 3 children who were older than me, and so they became babysitters for me. I was a 13-year-old, and they were 17, 18, 19. They were up there. I did not learn to speak Spanish because my father could not speak Spanish, and he didn't want me being able to talk to my stepmother and saying things that he didn't understand. There was one unfortunate reason for that as well. My father was an abuser.
Oh.
I learned from my biological mother, and I saw for myself what he did to my stepmother, number 4, mother number 4. And so as a result of that, I developed an interesting attitude toward women. When my mother disappeared, I sort of felt a negative feelings toward her, and my grandparents, who were my father's parents, and tried to reinforce that. So there was some kind of conflict in the family about that. So I had a negative feeling toward women.
And anger.
And then seeing what my father was doing to my stepmother, it went the other way. I said, that he's mistreating.
Yeah.
You don't do this sort of thing. By the way, I was also pretty religious by that time because of the training under my grandfather. And that's the way we started off in Santa Barbara. We then moved to Bellflower. And in Bellflower, I finished off junior high school, and then I started high school. And for the first time in my life, I actually went through a school from beginning to end. The first time that happened. Yes. and that was a great thing. And I just tried to get involved in all sorts of things to get as much as I could out of the experience. By that time, I still had not developed anything you could call a real interest in science. I played around with some invertebrates. I liked to capture fireflies and things like that as a youth, but I was not really what you'd call scientific material. And as I said, there wasn't a role model, and nobody had gone to college. I had an uncle, my grandfather's oldest son, who started pharmacology school at the University of Illinois just before my grandfather went bankrupt from the Depression. Oh. And that killed his chance. So there was nobody in the family that had an education beyond high school, and nobody in the sciences at all, anything that was remotely connected with the sciences. So I still had no role model.
With these changing environments that you were finding yourself in, your grandmothers, your fathers with the different wives and stepmother with the children, do you think you were an observer, though? Do you think you were wired to observe around you in that?
Well, yes. Actually, there was so much uncertainty that I had to look for opportunities to get stability. And I did develop a kind of a stubbornness. I would stick to a project, and if something went wrong, I would be very loath to drop what I was doing. Now, sometimes I wasn't doing something that was right, so this did not always work out the way it should have worked out. I just had to learn how to judge what was right and what was not right. I was fairly religious at the time, And I did gain some feeling of certainty from the religion.
And did you attend temple regularly with your father, living with your father?
He was a tenor. He was a cantor, not by formal training, but he had a good voice. The aunt that died with lupus was a soprano. She was in training as an opera singer. And my father, he was actually the black sheep of the family. It was difficult to get him to stick to anything, but he loved to sing. He had a very good voice, and he became the cantor at the synagogue in Bellflower that we went to, and that probably is what kept me going to synagogue, because he was the cantor there. So there was that. But for him, the religion was just a means to express himself as a cantor. He wasn't religious. You know, I was the religious one, and my grandparents were taken with that notion that, in a sense, I was the mechanism by which they could keep their black sheep under some control, with some connection to the religion, because my Mexican-American stepmother was not Jewish.
Right.
Neither was the one that preceded her, but when I went at 11 years old, I spent a very short period of time with her, and I don't remember pretty much about her. But she was she marked the first time that he broke with his religion and married a non-Jew. And so he was already on the blacklist with the family for that. And so my stepmother, my third stepmother, was just an addition. She just added on to that. So I went to high school, and I wanted to really fit in. So I went out for athletics, and I involved myself in any activity that I felt I could do something decent in. I was a sorry case in football. My great accomplishment was bloody noses and a concussion. But I found a home in cross country. I became, it fulfilled the need to be a more complete person than just a student, because this commitment to make high school my home base and something that I was devoted to, some source of certainty, made me more of a student, and I became a serious student in high school. But I didn't become committed toward any direction until I met my biology teacher. And as a sophomore in high school, I met Mrs. White. And she is what made me a scientist. She was such an inspiration, and she knew how to handle me. And, any of my tendencies toward being a little wild and disruptive, she was stern about it, but she was encouraging, constantly encouraging. Not the kind where she pounded me over the head, but it was kind of a soft encouragement. And I decided I want to be a biology teacher. And I just immersed myself. Biology was the class for me. The rest of the classes were okay. I'll attend them, but I couldn't wait for biology. The dissections and all the things that we did in biology, they were the height of the experience from being in high school.
It's amazing the power of a good teacher has on a youth, isn't it?
Yeah.
And
both ways.
Yes. I was going to ask you, how did you navigate? You knew you wanted to go to college, but from your bio, you were the first one to go to college. I imagine that your father may not have been too dialed into the fact that, getting my son in college might not have been his priority. How was it the biology teacher that helped you navigate that process?
I decided I wanted to go to college and become a biology teacher, and my father didn't know anything about the process. And I also developed an interest in soil microbiology. I found a book in the public library in Bellflower on soil microbiology, and I started looking at universities that had agronomy programs. But then I learned that you had to get together a little bit of money in order to go to a university. And I found that I would have to go to university either in California or Illinois. And then I learned that if I could be classified as a resident of Illinois, I would get a break on the tuition. This required that I communicate with my mother who lived in Illinois, and she could establish my residency. At that time, that was sufficient. And so with the help of my relatives in Chicago, I was able to get a letter from my mother stating that I was a resident, and I was able to go to University of Illinois as a resident of the state, even though I hadn't lived there for quite a while.
So you're an undergraduate at Navy Pier?
Mm-hmm.
And tell us about your experience there. What were you contemplating? You were going to be a biology teacher. You had agronomy. What you're kind of searching here for?
Yes, I was kind of open. I mean, the biology teaching was there, but agronomy had a specific subject matter interest to me. I was just attracted to it. The microorganisms. I was excited by microscopy. You know, the microscope was I just loved sitting by the microscope and just discovering things in the microscope. Anything I can pick up and look at under the microscope was just, you know, just heaven to me. So I started off sort of in the general science area. I thought that I'd got a good training at Bellflower High, but I discovered that no, I had some lacking. So as my first year as an undergraduate was sometimes rough, especially French.
Oh.
So it was not, I was no sterling student at the time. And so I struggled a little bit, and finally I got straightened out a little bit. And then I sort of thought, well, biology teaching is probably something that I'd rather do anyway. So I signed up for the biology teaching program at Illinois, which dictated at that time a strict curriculum of taking physics, taking chemistry, so, you know, lots of science courses. And the atmosphere at Navy Pier was very conducive to studying. Navy Pier was literally, for those that aren't familiar with it, a pier. Okay, it sticks out into Lake Michigan, and it's split down the middle, longitudinally, into a pier, which unloads ships, and a school, a university. And to get from the auditorium, which was out at the end of the pier, to physical education classes, which is at the base of the pier, it was called the 5/8 mile run. You know, if you had two classes that were one right after another, and they were at each end, you had to run the hall. But the important thing about Navy Pier was that a very large fraction of the students were veterans. Many had families. They were serious students. This was not a play school at all. When they held a dance, the hall was empty.
Oh.
I mean, you hear echoes. You know, and the dances were held in the auditorium. And so the teachers, it was a fairly young school. There was only 2 years of the University of Illinois, and that was politics because the downstate people did not want Chicagoans, which made up 4/5 of the population of the state, to spend all 4 years spending their money at a school in Chicago. They wanted that money to go downstate. So they limited it to 2 years. And the faculty that was gathered was not typical university faculty in the sense that they had all the security and the tenure of a 4-year school. These were moonlighters from the University of Chicago. So we got some pretty good teachers, okay, but they were serious as well. So we had serious students, serious teachers, and good qualified teachers and very capable teachers. It was a rigorous experience. And my second year, I took zoology, and that was my meeting with my first mentor, Professor Schumer. And I just flourished. I just loved zoology. I was just amazed by the variety of life, okay, and this is just animal life. And I just reveled at each phylum that we covered, and it was an epiphany to me. I just loved it. And at the same time, I was starting to move away from I had already started, but I was moving away from my religious orientation to some extent. It was becoming somewhat neutral, not agnostic in the total philosophical sense, but more in that direction. So I was becoming so interested in science, and I looked for opportunities to work for every member of the biology department I could. I mounted plants. I killed my first bird with a gun.
Oh.
I was the ornithologist of the department. We went out to collect birds. He was doing a study on bird migration. So I learned some things that I didn't really use later in life, but this gave me an idea of what research, involved.
Right.
So I was getting into science more and more. The 2 years were up, and so I had to go downstate. I worked summers as a relief elevator operator in Chicago in order to make the money to pay for school. I lived with my grandparents, so that took care of the room and board while I was Navy Pier. But of course, going downstate, I had to have enough money to pay for room and board downstate, and that came from my job as an elevator operator in the summer. So I went downstate, and that's when I discovered the difference between Navy Pier and the Downstate University of Illinois. My impression, the shock was I said, this is a play school. Where is there serious learning going on here? It was a flip side. It was like going to two completely different schools, and the temptation to get involved in all the fun-loving activities downstate was enormous. But I was determined to stick to my own goals and to try to maintain my education. And I still wanted to become a high school biology teacher. But I guess there's a little bit of rebel in me. I knew I was different, and this difference expressed itself in my attitude toward how student teaching ought to be experienced by a future teacher. I had decided, I sent for a journal that was published in the state of California for administrators of high schools about curriculum. And I decided from reading this journal that the teacher training approach in California made more sense. A teacher in California that's in training teaches one class for the entire semester and becomes totally immersed.
Right.
Okay, and the class is monitored by a regular teacher who is a training teacher, and you get to know the students very well. In Illinois, it's about 3 or 4 weeks. You spend teaching a whole day of school, and that made no sense to me. That made no sense to me, and so I decided not to enter the student teaching program. I didn't sign up for it. The dean of the School of Education called me in. And I was sitting in his office, and he said, you haven't signed up for the student teaching program. And I said, well, I prefer to wait until I get to California and go to get my master's and do my student teaching in California. He said, you can't do that. You can't maintain yourself in the biology curriculum. You can't be a biology teaching major and not sign up for biology teaching, student teaching. We will not allow that. So he kicked me out of the curriculum.
Oh.
First time I've gotten fired, okay? And so I had become a zoology major. So I converted to a zoology major, and then I had to fill in with straight zoology courses, and that was lots of fun. Ecology, and I took invertebrate zoology, and that's where I met my second mentor, John Corliss,
Yes.
who was a protozoologist. And the invertebrates, I already had a little bit of imprint on invertebrates from my youth, liking to collect. It's mainly insects because not all the vertebrates come to the surface of the soil
Right.
that you can see. And he was a protozoologist, so that was the invertebrate that I got kind of attracted to. And I started to look at all the invertebrates. I met tardigrades. That's where I met the tardigrades. And so many years later now, I see it, the tardigrades are becoming a very popular thing.
They're very fashionable these days.
Yes,
T-shirts and
it's so great to
memes.
see that.
Yeah.
So and I took a vertebrate physiology, and I enjoyed that. And then I finally graduated from Illinois and was thinking about getting hooked up with somebody at UCLA. I talked to Carlos, Professor Carlos, that talked about protozoology programs, and of course I was going home. I was going to teach in California, so it had to be a California school that I was going to because I was going to get my teaching degree there. And so I got the name of Theodore Jan. Okay, and he said, well, if you're going to go to a California school, especially in the Los Angeles area, you should go to UCLA, and I'll give you a recommendation and get Theodore Jan as your mentor. And so I graduated, and then I signed on to UCLA. Okay, and I became a member of the protozoology program, protozoa and physiology, whatever, at UCLA. And Ted Jan was a synthesizer. One of his inspirations was his ability to scan the literature and look at disparate papers that had come to unclear conclusions. These authors had gathered data, and they'd come to some just minor level of a conclusion. You know, this happened, and it was what I expected to happen, but they didn't go very deep. And he would synthesize the results of the various papers into something approaching a theory, and that had to do with transport of calcium, you know, and the membrane phenomena that had to be overcome or that had to be implemented in order for the transport to occur. And he became fairly famous for being able to do that. He became well known as a synthesizer. He worked a lot with physical chemists. Okay, and so a lot of the ideas that he got were not available to biologists who didn't think so much in terms of physical chemistry. And so that was an approach that was kind of appealing, and I stored that away in my mind. It's good to be a synthesizer, but I was still going to go and be a biology teacher. And so I finished my biology teaching training, and I met my next mentor, who was a training teacher. And he taught me what teaching was. What I had experienced at UCLA was not teaching, it was lecturing.
Ah, and who was that mentor?
George Bonoris. And that introduced me to the Greek community, by the way, of Los Angeles, because George Bonoris was Greek. And so I developed a ton of Greek friends. It's easy to do with the Greeks because they're very friendly.
Everybody's family.
Yeah. And so there was that. But I also learned that I'm not as good a teacher as George Bonoris, and the distance between me and George Bonoris, it was pretty large. Even though I was able to somehow convince the people at the school I taught at that I was competent enough that they made me a master teacher, and I began training teachers myself, I still knew the difference. And there was one teacher or a lecturer that was so good that he was a teacher. This was an embryologist by the name of Sheckman at UCLA, when I took my graduate work for my master's degree. So I knew that there could be lecturers who were so clear, who were so good, that they grabbed your attention and you learned from them. You didn't need the tools that a high school teacher needs, because you don't have to be in a college class, but you do have to be in a high school class in the public schools. So a high school teacher has to take students who don't want to be there particularly and make them want to be there. That is an ability that is really difficult to develop. You know, I had minimal ability in that area compared to George Bonoris. I mean, he was a finalist for Teacher of the Year in the state of California. I mean, he was quite good. I I'm glad to have mentors who are so talented that I have to learn from them. There's so much there to learn.
Yeah.
So this is great. So I like being with people like that.
But you did work for the Los Angeles City School, too?
Yes, I did for
Yeah.
4 years. I was at Hamilton High School, and I taught teenagers. So,
And how was that experience? Was it,
well, I knew already from the training of George Benoris that you can't walk into a high school classroom and start lecturing. This is not teaching. This is not teaching. You have to engage those students. And so that made a big impression on me. But it also made me think about what the difference is between the teacher in the classroom and those in the administration that are supposedly judging their teaching ability, particularly if the people in the administration don't have themselves a lot of experience in the classroom. This drove me, actually it resulted in an episode in my teaching career that changed the rest of my life. And that was George Benoris had a lot of great ideas for the curriculum, for the biology curriculum in the Los Angeles City Schools. The people downtown essentially ignored him because he did not have an EDD or PhD. Nobody called him doctor, you know. He didn't have that, and that's why they ignored him. That struck me as being
elitist?
counterproductive. Okay, I don't want to use any nasty words, but it struck me as being counterproductive. And it disturbed me very much. And that, together with the knowledge that I was not going to be a great high school teacher, made me decide to listen to Professor Jan, who had sent me a letter. He said, you know, you passed your preliminary exams at the PhD level. And you have a limited time where you can start your PhD training, and if you don't start it within that limited time frame, then you have to take the exam all over again, because the knowledge that you were tested on in that exam, you know, it's old. So those two things came together, and I decided to go back and get my PhD. Okay, and so I went back, and with Professor Jan as my mentor.
At Caltech?
This was at UCLA.
This is still at UCLA,
I went back to UCLA, and then I graduated from UCLA. He proposed a problem to me for my PhD thesis that was bioconvection problem.
Okay.
And this was my introduction to physics and biology coming together. Now, I also, at that time, began to see that mathematics was going to be an increasingly important part of biology. So I started taking calculus courses. I took physical chemistry. I took things that would build up my physics background. And then I accepted that problem, and I took it on. Now, bioconvection is a phenomenon that occurs in some cell cultures, and it does occur to some extent in nature, where organisms gather near the top of a solution, and then, for some reason, then unknown, they start to fall, but they fall in a pattern. They don't all sink down together, but they fall in a pattern that looks like a net, like a tennis net, from the top. But what's happening is they're boiling, and I don't know if you're familiar with the Bernard cell.
No.
Bernard cells are what are formed when water begins to boil.
Okay.
Yes, the water circulates, and there are the different cells within the water. It looks very much like a Bernard cell, and a lot of people thought, well, this was some kind of a Bernard cell. When they started to use that interpretation, then I said, well, there's one way that we can eliminate the Bernard cell as a contributor, and that is to take these cultures and get in a cold room where there's no heat transfer taking place, or you keep it down to a minimum. And another thing that could help us understand the mechanism, I began to think that gravity had to be important as well, is to eliminate gravity. Okay, now I was able to eliminate the influence of the Bernard cell in the cold room, but gravity was another challenge, and that was one of my first failures. I decided that if I could I had a camera mounted on a a microscope structure that looked at the culture, and I took it into a Piper Cub. It was a model of Piper Cub. It was a single-engine plane, and we went through what are called Garantaball arcs. These are arcs that are instituted in order to create microgravity, free fall. And that's what we did. Well,
Who owned the Piper
lighting,
Cub? Who was the pilot here?
well, I wasn't the pilot. No, I was in the back. I was in the back. So the lighting was not sufficient to get me good pictures. So I didn't get the data. What I did get was a green face as I got off the plane. One of my graduate school buddies, I got off the plane. He says, "Why is your face green?" And I didn't want to repeat that. I thought,
There weren't any more anti-gravity studies then?
No. Although years later that Jan wanted to get on to one of the satellites, not satellites, one of the rockets that was going up. But this was a project that wasn't going to sell to NASA. It would be a benefit for research, but it wouldn't sell. So I completed my work for the PhD, and I still did not have the mechanism clear. I knew gravity drove it, and I was convinced that it was not a thermal convection phenomenon. And I went to Caltech after Professor Jan talked to Charles Brokaw, who's well known in flagellant physiology and locomotion. He talked to him, and he recommended me, Brokaw recommended me to Theodore Y. Wu, who was in engineering science. So suddenly I was in engineering at Caltech. I was a postdoc. And the story of Caltech now becomes an important story for physiology as well, but it is particularly addressed toward bioengineering. I was kind of lucky. I dropped into a situation that was at the beginning of bioengineering in this country. In 1967, 68, around in there, Y.C. Feng, who is considered the modern father of biomechanics, and my mentor, T.Y. Wu at Caltech, tried to get the engineering division to start a bioengineering department at Caltech. There was at that time a member of the department, a member of the division by the name of Harold Wayland, who was very interested in doing intravital microscopy on cat mesentery microcirculation.
Oh.
And he supported them, but they were the front. But Harold Wayland wanted to bring cats into an engineering building at Caltech. There was an aerodynamicist, fluid dynamicist, I won't mention his name, whose wife was so against the idea of anything like a vivarium or even animals in an engineering building that he raised his voice and was one of the primary causes of that attempt to start bioengineering at Caltech to fail. As a result of that failure, now we're talking bioengineering history, as a result of that failure, Y.C. Feng left Caltech, went to UC San Diego. And one of the icons of this society, of the APS society, Xu Chen, can tell you what happened down there because he knows the full story. But they developed a bioengineering program down there that was the only one in the University of California system for many years, and it was referenced by much of the bioengineering world as being, especially in the areas of circulation, microcirculation in particular, in Tagliata, Xu Chen, Gerrit Schmidt-Schoenbein, I mean, so many people are there. And Caltech had only my mentor left behind, with Y.C. Feng left. So what was he going to do? Well, Harold Wayland somehow got the Division of Engineering to allow him to conduct his intravital microscopy experiments with cats in the building, which was Thomas Lab, which was where I was located. And he would get visiting professors and so forth who were biologists, because he was an applied mathematician. He didn't know anything about dealing with the animals. And they had cats in there. And his lab was next to mine. And I was hired to do what has become what I think is my most valuable asset. I'm a lab rat. I gather data. That's my value. I am not a great theorist, I'm certainly, I don't consider myself a great scientist. I'm a good scientist, but I gather data and I let the great theoreticians do with that data what they think they can do in order to develop a theory or something like that. Now, at that time that I was hired, there was in microorganism locomotion a problem that was called Gray's paradox. Gray was a British biologist who noted that if you tried to apply the law for movement of objects in very viscous fluids, or that were very, very tiny, raindrops and things like that, if you would try to apply the law that's normally used for that to microorganisms swimming, the organisms couldn't swim. You see, the organisms did not know that they were violating this law. They just swam. They just swam. So Gray presented this to all the fluid mechanicists, one of which was my mentor, Theodore Y. Wu. And I was supposed to get the data that would allow any new theories, any new iterations of the developing hydrodynamics, which was termed biofluid dynamics by Sir James Lighthill. They were supposed to predict what a result would be if you plugged in real numbers to their equations, and that was supposed to match the swimming that I saw. So I took the movies, I did the analysis, and I gave them the data, and they plugged it in, and the papers came pouring out. It was just great for me. I was just thrilled by the people involved, and I loved that role. I was the guy down in the lab, and I just loved that role. And I was so elated when the first graduate student that got his PhD in this particular program had his PhD thesis published in the Proceedings of the Royal Society B, which was quite something.
Very impressive.
Yes, Alan Chuang, and that was really something, and I just felt so great about that. And it was also at Caltech that I started meeting some pretty influential people, important people.
And remind us what year this is?
I started at Caltech in '69, and so this was going on between 1969 and 1976, 77.
So a changing world, and especially a changing California, and exciting times in science too. The lay people were becoming very interested in science too. It's the '60s. And who did you meet there? That was.
I suppose there were two people that I met. They're both Nobel laureates, but one of them was Max Delbruck. But I didn't really meet him. I was a biologist, and here I was over in engineering. If I wanted to listen to a biology lecture, and there are tons of great lectures, so I'd go over to biology, which was at the other end of campus, and also Charles Brokaw was there, and he's the one that recommended me. I would sit and listen, and very often would not understand what the lecture was about, or there were many parts of the lecture. My attention was really drawn to the end of the lecture. And I would always sit close enough to hear Max. What he would do at the end of every lecture was he would start his question by summarizing the entire lecture in one sentence, maybe two. And that's what I went for, because then I knew what the lecture was about. Whatever I didn't understand the lecture, max at it. And the rest was just frosting on the cake, because that was it. And of course, this was a constant lesson in clarity. Okay?
Right.
These people had the ability to make things clear. They were able to process them in a way and make them clear. And the second person I met, actually, it was, they had these afternoon teas. And new faculty, it's kind of crazy, but at that time, postdocs were members of the Caltech faculty. They were the absolute lowest rung. The next step was the ground. And we'd go to faculty meetings. We had no vote, but I mean, some of the fascinating subjects that were being discussed by some pretty smart people, you'd hear it, and it's kind of training. If you got an academic job and you became a member of an academic senate, you knew something about how an academic senate works, at least Caltech style. But in any case, so they had these teas for the new faculty, and this guy comes up to me, and I recognize him, and personable. It was Dick Fineman, Richard Fineman, and what am I interested in? I mean, as soon as he found out that I was a biologist, he started going one of his pet peeves. He started asking me about psychological science. And I found myself having to defend psychologists. What do I know about psychology research? Of course, I had to take some psychology in becoming a teacher. But here I am trying to defend, not that I really wanted to, but, all I could do was, empirically, some of their results work. So they seem to solve a problem, and it seems to have some clinical benefit. But as to the neurological, biological underpinnings that they may be proposing, I'm in no position to defend that. Go see Sperry, the split brain guy. He knew Sperry, but he wasn't teasing me, because he was usually serious about these things. I also got to see him because he was good friends with my mentor, Ted Wu. Ted Wu, he's an amazing guy. He loves science, and you could tell it. He's so enthusiastic, so engaging, and just a sweet guy. That if you don't love Ted Wu, you have no feeling. It's just that way. Everybody at Caltech just that knew him. It's a small school, but there's some people that other people don't know. It happens. Yeah. And so Ted Wu would be talking to Fineman at lunch or whatever, and he started talking about some of his stuff, and just Fineman became interested in the data. Okay, and there I was down in the lab, so he'd come down and talk about the data.
The data guy.
Yeah, the data guy. And it was fascinating. I mean, he had some chuckles. I have ways of expressing things that left over from my school teaching days, I talk about the rhythms of biology, and he got a chuckle out of that. Of course, that may have been also because he plays bongos, and he's very much into rhythm, But it was that kind of thing. But what I learned from him was that the personalities weren't important.
Right.
It was the problem, and you focus on the problem. And he would always talk about the problem. He never got political with me. I mean, and there are other things that I learned from him, not directly, because we were only talking about the data and sometimes psychology, but mostly the data. And it was a challenge. He found it interesting because first you have Gray's paradox, in which the Stokes equation, which had been useful for so many people in fluid mechanics at what we call low Reynolds number, had to be modified. And the modifications by applied mathematicians, I was just surrounded with applied mathematicians. And all of these modifications help make the data better be predictive of the model. They had to make the model so that the data could be plugged in and it would predict the actual behavior of the organisms. So they improved, the additions. They did, and it would be often by mathematical tricks. Now, I had calculus up to partial differential equations. So I had an inkling of what they were talking about in many of these seminars. But very often they'd introduce an approximation, or they'd introduce a correction factor that was itself another equation, and I just do a double take. How come they're introducing it at this point, and how do they know to do it? And the applied mathematicians, some of them were kind of a little bit impish, shall we say. They would tease me, you know. And one in particular is a professor at Caltech. You know, he always, you know, would say, it's trivial. You know, read something. So, you know, and that was the atmosphere, and there are many things that I learned from Fineman, not necessarily directly, because I wasn't part of any intergroup, you know, but I learned from his writings. And I went to his lectures, and again, like Delbruck, his lectures are so clear that you walk out of the room saying, I understand quarks. 12 minutes later, after you've asked yourself, but what? Not really. But he was so great at that, at the clarity. He was so clear, this ability to make things simple.
So it sounds like there was an atmosphere of inclusion. The postdocs got to be in the faculty. There weren't so much the sense of fiefdoms, and so this really sparked collaboration and the ability to talk across discipline and go deeper into and expand, just to be able to explore different areas for you. But
yeah, Caltech's a special place.
You didn't stay at Caltech. Where did you go after that?
Well, there were students of Wu who graduated, and they went on to other careers. There was one, now just to put a little negative aspect onto what you just said. In general, Caltech was just a great place, but they're humans, okay. One of his students talked Chinese enough so that he had an accent. But he was, this student I talked about, that his thesis was published in the Procedure. A brilliant guy. He was hoping to get a faculty appointment at Caltech, and it didn't happen. You know, the years went by, on and on and on, and there was a comment that somebody on the selection committee made that got back to him about him not being clear enough in his lectures. You know, I wasn't in a position, you know, I'm not a fluid mechanicist, so it's not for me to judge. So so anyway, he left and became head of the Hydraulics Institute at the University of Iowa. So the feeling was, people need to move on. So I decided to move on. But I should not forget, was probably my most significant scientifically achievement at Caltech. Part of this atmosphere of openness involved your being able to go down and just sit in any class. And I went to a class, HY 101, taught by Frank Marble, just to learn a little bit about fluid mechanics. And he gave a lecture that included a description of a physical phenomenon, and as I was sitting there, I said, that's it. That's the mechanism for bioconvection. And I got so excited, I ran upstairs after the lecture, and Ted Wu was out of town. But I had to tell someone, and the department I was in was called Engineering Science. There were only about 2 in the whole country called Engineering Science departments. And it consisted of 2 faculty members, Ted Wu and Milton Plessett, who was a nuclear plant heat transfer guy, but he knew fluid mechanics. And I blurted out my interpretation. And he said, yeah. He said, yes, that makes sense. And the next thing I knew, we had a paper in Nature.
How wonderful.
It's Plessett and Winet, because he's senior. But that finally, there was a theory. So I have a theory that is the only real theory in my entire career, and it was back in 71.
Wow.
It's been downhill ever since.
What was going on in your personal life during this time, your family dynamic?
Well, I think this is an event happened that probably severed my last religious connection. I lost my daughter at the age of 4. And I can't say it was the same thinking that Darwin, when he lost his Annie, but that kind of completed the severing for me. And so then I left Caltech, was after that, and went to Southern Illinois University on the faculty there. I joined the department, and this was a physiology department, okay? And I had some adventures there. I learned a lot about faculty politics, and I also learned that my son was allergic to the flora in that part of the state, and he developed asthma. And as part of the faculty politics, I was overruled. I was the head of a search committee, and I was overruled by the dean of the College of Arts and Sciences. And the head of the department backed me up. He said that this is something that, as a search committee, they're supposed to have freedom to do this. It just so happened that the person that our committee selected was from India. I'm not sure that had anything to do with it, but it was unacceptable to the dean, and the war started between the head of the department and the dean, and so there was that stress, and then my son having the asthma, and my wife wasn't that happy about it. My wife's a hematologist, and she had quite a career of her own back at Orthopedic Hospital. Although the medical school was ready to accept her, they were going to give her practically anything she wanted. But Southern Illinois, Carbondale was not the best place. So I said, okay, I'll change careers. So I took a job actually at her hospital, at Orthopedic Hospital, and I had to switch careers. And then I became a bone physiologist. I had to learn bone physiology, and the career went on from that. I finally became a member of the Department of Orthopedics at USC, University of Southern California. And by the way, I got my first NIH grant when I was at Southern Illinois. And I had to end that grant. I had to finish it off. Actually, I had the help of Ted Wu. He let me have a lab at Caltech. And I finished that off, so that stopped my career with ciliary locomotion. And I had to start a new career, and I was looking around for something to do, and there are people in the hospital that had all kinds of ideas of what I should do. And I was really struggling. And then I met Fred Rhinelander. Okay. And Fred Rhinelander introduced me to vascularity in bone. And I remembered Harold Wayland, and he introduced me to some people who had been studying real circulation in bone, and that was Albertson and Bronemark, these are big names in dentistry. Not too many people necessarily know about them, but they did intervital microscopy. And I got a Fogarty fellowship to go to Göteborg and learn intervital microscopy, although I knew a little bit about it from Harold Wayland, but I learned it with bone, from these two people. And I came back, and then I submitted an application to NIH, and surprised the hell out of all the orthopedic people, because they saw this lost guy. I mean, this guy didn't know anything. He'd lost, but I had something. This was microscopy, okay, and I had learned a technique, and so I got my first NIH grant. And suddenly I had all the things I wanted at Orthopedic Hospital. They gave me a lab and everything like that, because it was a big deal. Very few of them had ever had anything like an NIH grant. So, and that was my career at USC. I continued on that way until I started thinking about implants, because bone is a lot about implants, studying bone. And I went to this meeting where I met an implant expert by the name of Jeff Hollinger, who happened to be at Walter Reed. We struck up a friendship, and I told him that I'm interested in studying implants and putting them in my little bone chamber. I'm not going to have time to talk about it, but it's a chamber that you implant and you look through it. It's in the bone, and you can actually see the bone grow in and see the vessels grow, and you see microcirculation in the bone.
That's neat. We can link to it in the video, yeah.
Okay, and then so I learned about that. I didn't like the design of the Brånemark Albertson microscope because it was vertical, and it pinned the leg down, and I thought that that's compressive, and so I invented a horizontal intervital microscope, and I got Harold Wayland to help me with the optics because he knew optics, he knew intervital microscopy, and so that was the first horizontal intervital microscope, my invention. I've never patented it, and then I started to use that on the rabbits, and then I started publishing papers, and after I met Hollinger and I went to Walter Reed for a couple of years to learn about the polylactic polyglycolic acid implants, I started putting that in the bone chamber and studying that, and that took me to the time when the second time I got fired.
Uh-oh.
The orthopedic hospital decided to reaffiliate themselves from the University of Southern California to UCLA. And the University of Southern California got extremely upset by that. And they fired all of us. We were on the faculty at USC. They fired all of us because they fired everybody that was associated with orthopedic hospital because we were cross-appointed hospital and USC. And USC, part of the agreement with orthopedic hospital and UCLA was that they would pick us up. So that's how I got back to my graduate alma mater, as a result of the hospital reassociating themselves. And that UCLA, it was a little, we were just there. And so we had to conduct our work. And the last few years of my life was mostly teaching. I got acquainted with people in bioengineering at UCLA. I had been in bioengineering and orthopedics at USC. And so I had courses that I taught at USC that I could help teach at UCLA because UCLA was just developing their bioengineering program. So I fit in there, and it was biomedical engineering at the time. I fit in, and gradually I became a member of the bioengineering department, and to add on to the Department of Orthopedics at UCLA. And I spent my time at UCLA almost exclusively in the bioengineering building, not in orthopedics, because they didn't really know what to do with the research people from orthopedic hospital that went to UCLA.
What, in the short time that we have left, what advice would you give to students? It's such a different environment now. I think the transition from, in high school to undergrad and know what you're going to be, when you're in second grade. What advice would you give to students now who are considering a career in science?
My life has been a lot of change and a lot of uncertainty. And this would be kind of general advice that would really go beyond science. Make yourself useful. I'm a lab rat, I generate data, okay? I don't have any great ideas, that get people excited. And that's a role that I love, and you have to become good at something, okay? And make sure that you do it the best way possible. And that won't guarantee anything, but that dependability is itself a value. It is a talent itself because there's a lot of undependability out there. And so it'd be that general thing because there are so many times when life changes and it forces you to go in a direction you didn't expect. You've got to be prepared for that, but if there's something stable that you can keep, that's you, okay, that anybody can refer to and say, oh, that's so-and-so, okay, he or she is good at that.
Right.
If you've got that, then you've got something to sell. Otherwise, you are always on the short end. You're asking for something instead of having something that somebody else wants.
Well, thank you so much for being so gracious with your time and sharing your personal story and your academic story. You've enriched all of us, and I just thank you.
Thank you for your interest.